Antibiotics have revolutionized modern medicine, enabling doctors to effectively treat bacterial infections and save countless lives. The history of antibiotics dates back to the 1920s, when Scottish scientist Alexander Fleming discovered the first antibiotic, penicillin. Fleming was working with bacteria in his laboratory when he noticed that a mold called Penicillium notatum had contaminated one of his Petri dishes and had killed the bacteria around it. Intrigued by this observation, he isolated and purified the substance produced by the mold that had this antibacterial effect, which he named penicillin. However, it was not until the 1940s that penicillin was mass-produced and used widely in medicine. During World War II, the United States government invested heavily in the production of penicillin to treat wounded soldiers. By 1944, enough penicillin was being produced to treat all Allied forces. Penicillin was a game-changer, reducing mortality rates from bacterial infections dramatically. Since the discovery of penicillin, many other antibiotics have been discovered and developed. These antibiotics work in different ways and are effective against different types of bacteria. Antibiotics are drugs that are used to treat bacterial infections. They work by killing bacteria or preventing them from reproducing and spreading in the body. Different antibiotics work in different ways, but the most common ways that antibiotics work are inhibiting cell wall synthesis. Some antibiotics, like penicillin, work by preventing the bacteria from building a strong and stable cell wall. This makes it easier for the immune system to attack and destroy the bacteria. One way that antibiotics work is by inhibiting the synthesis of bacterial cell walls. Bacterial cell walls are essential for the survival of bacteria because they provide structure and protection against external factors. However, not all cells have cell walls. For example, human cells do not. Antibiotics that inhibit cell wall synthesis, such as penicillin, work by blocking the enzyme that bacteria use to build and maintain their cell walls. This means that the bacteria are unable to produce a strong and stable cell wall, making them more vulnerable to attack by the immune system. Specifically, penicillin and other beta-lactam antibiotics work by binding to and inhibiting the activity of enzymes called penicillin-binding proteins that are responsible for building the bacterial cell wall. Without functional PBPs, the bacteria are unable to synthesize new cell wall material, and their existing cell wall becomes weaker and more susceptible to damage. Because human cells do not have cell walls, beta-lactam antibiotics like penicillin are relatively safe for humans to use. However, some bacteria have evolved mechanisms to resist beta-lactam antibiotics, such as producing beta-lactamase enzymes that break down the antibiotics before they can reach the PBPs. To combat this resistance, newer generations of beta-lactam antibiotics have been developed that are less susceptible to beta-lactamase degradation. Inhibiting protein synthesis, other antibiotics, like tetracyclines, work by preventing the bacteria from making proteins that they need to survive and reproduce. Another way that antibiotics work is by inhibiting protein synthesis in bacteria. Protein synthesis is the process by which bacteria produce the proteins they need to survive and reproduce. Antibiotics that inhibit protein synthesis, such as tetracyclines, work by binding to the bacterial ribosome, the cellular structure responsible for producing proteins, and preventing it from functioning properly. This prevents the bacteria from producing the proteins they need to grow and divide, ultimately leading to their death. Tetracyclines and other antibiotics that inhibit protein synthesis are effective against a wide range of bacterial infections, including respiratory infections, skin infections, and sexually transmitted infections. However, they can also have side effects in humans, such as upset stomach and sun sensitivity. Damaging bacterial DNA, certain antibiotics, like ciprofloxacin, work by damaging the bacteria's DNA. This prevents the bacteria from replicating and spreading. One way that antibiotics work is by damaging bacterial DNA. DNA is the genetic material that bacteria use to store and transmit their genetic information. If their DNA is damaged, they may be unable to function properly or reproduce. Antibiotics that damage bacterial DNA, such as quinolones, work by interfering with enzymes that are essential for DNA replication and repair. This can cause breaks in the bacterial DNA, which can ultimately lead to the death of the bacteria. Quinolones and other antibiotics that damage bacterial DNA are commonly used to treat urinary tract infections, respiratory infections, and other bacterial infections. However, they can also have side effects in humans, such as nausea, diarrhea, and dizziness. It is important to note that antibiotics only work against bacterial infections and are not effective against viral infections such as the common cold or flu. Additionally, antibiotics should be used only as prescribed by a healthcare professional, as overuse and misuse of antibiotics can lead to the development of antibiotic-resistant bacteria. Thanks for watching.